Think Forward. Think Research Channel.
It's my great pleasure to introduce my good friend and fellow intramural scientist, Steve Straus, who will be giving today's NIH Director's Lecture. Since it is an alternative Wednesday, we thought it appropriate that this lecture be on a Monday. Dr. Straus was appointed the first director of the National Center for Complementary and Alternative Medicine in October 1999. An internationally recognized expert in clinical research and clinical trials, Dr. Straus is also chief of the Laboratory of Clinical Investigation at the National Institute of Allergy and Infectious Diseases. He has extensive experience with conditions that have alternative remedies, including chronic fatigue syndrome, Lyme disease, AIDS, chronic hepatitis B, genital herpes, and chronic post-herpetic pain. His scientific work includes studies of the molecular biology, pathogenesis, treatment, and prevention of viral infections and immunological diseases, resulting in the publication of over 350 research articles. His accomplishments include showing that acyclovir suppresses recurrent genital and oral herpes. Dr. Straus also is a key player in the characterization of autoimmune lymphoproliferative syndrome, or ALPS, a genetically determined disease. He has been a distinguished colleague and friend to many in the audience and has played a critical role in invigorating clinical research at the NIH and has chaired a committee which produced a report which we know affectionately as the Straus Report. Dr. Straus earned a BS degree from MIT and an MD from Columbia University College of Physicians and Surgeons. He joined NIAID as a senior investigator in 1979. He is an elected member of the Association of American Physicians, the American Society for Clinical Investigation, and has been appointed to the editorial boards of several scholarly journals. He's received 5 medals and other commendations from the U.S. Public Health Service, including the Distinguished Service Award for his work on clinical research at the NIH. His topic today is exploring the scientific basis of complementary and alternative medicine. Please join me in welcoming Dr. Straus.
Thank you, Michael. It's a great honor. And in my years here at the NIH, I had hoped that I would have the occasion to give a director's talk and imagined not only that it would be on Wednesday, its proper place, but that it would be on a subject that I had studied for many years, had mastered somewhat, and had made some original contributions. Today I'll be talking about an area that I am still beginning to learn and to which all the contributions have been by other people and my colleagues. And the challenge for me is to try to summarize the things that I feel exciting about the field in exploring the scientific basis of complementary and alternative medicine. I want to start with a few definitions and give you some backgrounds to the field. I'm talking about a series of diverse medical and healthcare practices that share the fact that they are outside the realm of conventional medicine and are yet to be validated by scientific methods. These include things that we call complementary because people choose to use them in addition to the kinds of practices that we have long sought to develop and champion here at the NIH. And alternative practices are ones that people choose in place of those remedies, however effective they are. Now, these are truly diverse areas, and we aggregate them for work purposes at the National Center for Complementary and Alternative Medicine into 5 major domains. These are biologically based systems, including special diets and thousands of different kinds of herbal remedies. These include manipulative and body-based systems such as massage, chiropractic, osteopathic manipulation. Energy therapies involving things that are truly based on energies with electromagnetic radiation, such as magnets and electroacupuncture, and things that are purporting to work through energies, such as Reiki and Qigong and other forms of of Asian healthcare practices. There are mind-body approaches, of which many are within the field of CAM, including yoga and meditation. And there are whole systems that exist as parallel healthcare universes we call alternative medical systems, some of which are ancient Ayurvedic medicine from India, traditional Chinese medicine, as well as things in the past 2 centuries that evolved in Europe and the United States, such as naturopathic medicine and homeopathy. Now, these are enormously popular practices, and while the specific data here might be questioned, the survey by David Eisenberg at Harvard, published 4 years ago, suggested that people in the United States are making more visits to CAM practitioners, and that that number of visits grew more rapidly in the decade of the 1990s than to mainstream conventional MD practitioners. This is big business. You have to wonder what it is that drives people to choose these therapies when scientific medicine has wrought such extraordinary achievements in prolonging life and improving the quality of life over the past century and a half. And in part it has to do with things that some of us find unnerving or discomforting about conventional medicine, things that push some of us away. The fact that we can't cure all of the patients we would like to cure. That many of our therapies have adverse effects. They're not risk-free. We practitioners are bound to shorter and shorter increments of time we're allotted to visit with our patients. Managed care dictates that we crank patients through. Our healthcare process is highly technical rather than personal, and our care is fragmented among many specialists. In contrast to this, there are things about complementary and alternative medicine that people find attractive. There are, of course, media reports of dramatic results. There is the belief that I'll come back to that these treatments are natural and therefore not only effective but are safer for you. These approaches empower patients. This is a good thing. Our patients have responsibilities for their own care and making their own decisions. And CAM approaches emphasize not only the biochemical and physiological aspects of illness, it focuses as well on the spiritual and emotional consequences of that illness. Summing a lot of these things up, CAM therapists also pride themselves in the 3 T's: touch, talk, and time. And I'd have to say, as a physician who's practiced for 23 years in this building, I have to admit that I somewhat bristle at the idea that it's only CAM practitioners who could spend time with their patients and care about the whole patient. It's something that we are privileged to do on a daily basis here in the NIH Clinical Center. Now, what is the evidence for these things that draw patients towards complementary and alternative medicine that's made it so appealing? Well, the evidence in terms of the effectiveness of these practices is largely rooted in anecdotes and tradition, not in rigorous journal articles. Although there's beginning to be small studies, and as I'll attempt to show you, my colleagues and our many grantees in the growing community around the United States are doing a better job of developing evidence about efficacy. There's also growing evidence about safety. There is a concern about safety, not only whether there's an inherent toxicity, and most of these products are safe, but there are some we know are not safe. But there's a more pragmatic concern, even for safe things, that a person will choose to use them in place of an effective therapy, or in addition to an effective therapy, and in some way, as I'll show you, interfering with that therapy. It's this background that led to the establishment in the NIH a dozen years ago of an Office of Alternative Medicine and its elevation in 1999 to the National Center. And our mission, very concisely stated, is to conduct rigorous research on CAM practices, to educate and train a cadre of researchers who are capable of doing the kind of work to provide the evidence that the public deserves, and to take that information, to use it to raise the level of understanding about these practices by the consumer, and by health professionals. That's our goal. Now, we have to choose among the many priorities of how we spend the money that the American people generously give us, which is $105 million this year. And our system of prioritization is really the same as the rest of the NIH. We invest more in those things that have a greater burden on American health. We invest in those practices that Americans are using the most, and you'll see this in some of the research that I summarize. And as an investigator, I'm interested not only in knowing whether a practice works and whether it's safe, but why it works and what the underlying mechanisms might be. We invest more in things for which there's already evolving data rather than just anecdotes, although there are times to invest in those in an exploratory way. And we invest in those things that are ethical and feasible. Just because the public may choose to use a certain practice doesn't mean we can study it. We can't ask somebody to sign informed consent to withhold certain practices that we are bound through good evidence to rely upon. And in the NIH as a whole, we invest in those areas where private investment is lacking. And in this area, it's everything. Now, we have set out a series of investment priorities with that kind of strategy. Like most of the NIH, our major investments is in extramural research. We're funding 200 applications for research training and research around the United States today, as well as a few growing internationally. And these fund a whole range of approaches, phase 1 to phase 3, clinical studies, preclinical, mechanistic, and translational research, as well as research training. And here in the clinical center, under the able leadership of my colleague Mark Blackman, we are establishing a laboratory and clinically based research program. It's beginning right upstairs, and it's emphasizing clinical and translational studies of CAM approaches to age-related stressors. The frailty, the dementia, the depression, the loss of bone mass that occurs with aging. And that intramural program, as all intramural programs here, creates a nidus for research training. The end of our strategic consideration is that we invest today more in clinical research than in basic research. About 5/8 of our dollars is spent on clinical research because the public deserves answers sooner while we're finding out additional information, why some of these practices work, how they work, and what interactions they may have. This is sort of the exact mirror opposite for most of the other NIH institutes, which invest primarily in basic discovery. In addition to many exploratory and small studies, we have already mounted and are mounting a series of large multi-center trials that I believe are designed with the same kinds of rigor as any large trial that should lead to public approval and acceptance of a mainstream therapy or preventative agent. We have completed a study of St. John's wort for major depression that I'll discuss. It's in press and will be published next month. That study in collaboration with our colleagues in NIMH. In collaboration with the Cancer Institute and their their community-based cooperative oncology program, we're studying shark cartilage as adjunctive therapy for inoperable non-small cell lung cancer. With the Aging Institute, the Neurology Institute, and the Heart Institute, we are doing a study in 3,000 otherwise healthy aging Americans asking whether the preliminary data for ginkgo biloba is correct and that it can prevent dementia. With the Arthritis Institute, we have mounted the largest ever study of acupuncture for osteoarthritis pain. Also with the Arthritis Institute, we have a study of 1,588 subjects being enrolled in 13 centers asking whether glucosamine or chondroitin sulfate, alone or in combination, are superior to placebo and might be as good as the current main therapy, cyclooxygenase 2 inhibitors. And with the Cancer Institute, we're privileged to help support a 32,000 patient study for prevention of prostate cancer with vitamin E and selenium. With the Heart Institute, we are about to award a major multi-center trial for the first time of EDTA chelation therapy for coronary artery disease. And with the Diabetes Institute, we have called for applications to study two of the world's most popular herbal products for the treatment of prostatic enlargement, which many men in my age begin to consider as a health factor. Now, these domains of complementary and alternative medicine have vastly disparate levels of scientific evidence about them. I'm going to emphasize two primary areas today, ones that I think set the standard for where we may go, but understand that the data I'm going to show you is the best and not the average. And I'm going to begin by summarizing work that's being done related to herbal medicines. I mentioned our study with NIMH of St. John's wort. Let me give you some background and some idea of what we're trying to achieve here. This is a medicinal herb that's incredibly popular, with $6 billion of sales in a single year in Europe by this survey in 1998. It's used primarily for treatment of depression, other mood disorders, anxiety disorders, many other conditions. Like all plants, it's a complex mixture of many chemical constituents. We know that at least one of those constituents, hyperforin, contributes to its antidepressant action. And we have learned, as I'll show you through work done right here in the NIH Clinical Center, as well as smaller studies elsewhere, that St. John's wort, while it may be active in depression and other clinical conditions, also interacts with drugs, birth control pills, AIDS drugs, and drugs important for preserving the survival of transplanted organs. Now, it's the background suggesting that St. John's wort was effective for depression that led my colleagues in 1998 to begin to design a study that has been conducted and, as I said, completed. Let me give you a sense of the background data and how it evolves in this somewhat contentious field. A survey of all the research articles on controlled trials of St. John's wort for depression, published in the British Medical Journal in 1997, looked at the relative extent to which patients on St. John's wort did better than patients on placebo. This is the odds ratio. And you can see in this dozen studies, wide-ranging, with some studies purporting extraordinarily greater efficacy at a level actually unseen with any conventional antidepressants. These, however, were small studies. They were poorly designed with unclear endpoints and, in many cases, an unclear characterization of the form and severity of the depression of the subjects enrolled. Since 1997, there have been 8 additional studies, and what you can see in this graphic is that the average hint of effectiveness is now lower, because these studies have gotten progressively larger and progressively more careful. And it's what we know about doing trials of antidepressant drugs, as well as this background, that led to the design of the multi-center trial that Jonathan Davidson at Duke University chaired. This is a study with 2 phases, a 2-month phase and then a 4-month follow-up phase, in which patients were randomized to placebo or St. John's wort or one of the best existing contemporary antidepressant drugs, sertraline or Zoloft. Almost none of the prior trials actually had an active drug comparator. We will hear in the next few weeks, because this article is finishing peer review, how this turns out. But however it turns out, I believe it'll set a better standard for figuring out how to study complementary and alternative practices, because we've picked patients carefully and we've used rigorous endpoints. In this study, we've used the Hamilton Depression Scale as the primary endpoint and the change in that, and as a second endpoint, the rate of complete response, total amelioration of all depressive features in the Clinical Global Impression Scale and the HAM-D scale. This is the way you study antidepressants. Now, in this study, we were not using a synthetic drug. We were using a botanical extract. And again, as I come back to this notion that if it's natural, it must be safe and effective. Molière said it well 3 centuries ago. People can be induced to swallow anything, provided it is sufficiently seasoned with praise. And the praise has been unchallenged in this field, and that's changing. It needs to be challenged by evidence. Now, there are risks of these drugs besides the fact that they won't work. Here is a clever one from Glassbergen that reads, "It's said that St. John's wort may improve your mood, but perhaps it's time to cut back the dosage." One risk, of course, might be that you have too much of a medication. But a more important risk, certainly in terms of St. John's wort, was revealed in this very building by colleagues of mine in our AIDS clinic. Judy Falloon and her colleagues upstairs on the 8th floor surveyed AIDS patients and found that the vast majority of them use dietary supplements and other CAM approaches. She and Steve Piscatelli, a research pharmacist here at the NIH, took normal volunteers and gave them one of the potent new classes of AIDS drugs, the protease inhibitor, Crixivan or Indinavir, and showed that taking that drug established very good blood levels, and those levels at all times around the clock was sufficient to inhibit the replication of the virus, at least in the laboratory. But when they added to that very dose, the same dose of St. John's wort in the depression trial, this is what happened to the blood level of Indinavir. It fell substantially to levels that at many times of the day would not have been adequate to inhibit the replication of the virus. So you have to ask, if it's going to be effective, it will be effective because it in some way interrogates natural biochemical systems. And that produces the possibility that it will also have unanticipated risks. So the question is, why is St. John's wort doing this? We took this as a vehicle and as a signal to us in NCCAM to call for applications specifically to begin to study the potential interactions between botanical products and drugs. Now, when some of these herbs were first used by indigenous peoples 1,000 years ago, you didn't have to worry whether they interacted with birth control pills or Indinavir, but we do today. And this interaction is something that we're studying through grants to about a dozen applicants around the country, doing preclinical phase 1 and phase 2 studies. I want to give you a sense of where this could lead through some lovely work that's now being finished up and not yet published by Steve Cleaver of GlaxoSmithKline in Research Triangle Park, North Carolina. He reasons that interactions between herbs and drugs, like interactions we're learning between drugs and drugs, relate to the fact that these drugs interact with a basic chemoprevention system that we as animals have evolved over millions of years to have to protect us for things that we eat in the environment that may be dangerous to us. This pathway involves detoxifying things that are foreign chemicals as well as things we generate in our body, things we inhale, things we ingest, things that we absorb through the skin, and things that are synthesized in our own tissues, particularly the liver. And the detoxification pathway involves a series of proteins, oxygenases that oxidize chemicals, conjugation enzymes that attach sugars to them to make them soluble, and proteins that can now pump these solubilized substances out of cells, excrete them into the bile, into the intestinal tract, so you can get rid of them. We know that many drugs interact with each other and do so by dealing with some of the enzymes in this pathway. And Cleaver and his colleagues reasoned that one of the enzymes that St. John's wort may be dealing with is known as cytochrome P453A, or CYP3A. This is an enzyme that we know can oxidize a particular drug, promoting its excretion. Some of the drugs that interact and upregulate the activity of CYP3A include birth control pills and cyclosporin, this drug important for preserving transplants, as well as Indinavir. I'm sorry, these drugs are targets of CYP3A, and CYP3A activity will speed the metabolism of those drugs. Now the question is, what regulates the expression of CYP3A? It comes from a gene that's regulated by an upstream promoter, and various drugs can induce binding of nuclear receptors to that promoter, increasing expression of CYP3A, increasing the accelerated metabolism of these drugs. The kinds of drugs that we know can upregulate the expression of CYP3A include rifampin, the tuberculosis drug, steroids, diabetes drug, the anti-epilepsy drug phenytoin, the cancer drug Taxol, and possibly, and this was the purpose of this work, St. John's wort as well. Now, if this is true, and if St. John's wort can activate the expression of CYP3A, it has to work by inducing the activity of a nuclear receptor that would be expressed where CYP3A works. In the liver and intestine, it would have to bind to that promoter, and it would have to activate the clearance of endogenous or exogenous chemicals. That's exactly what Cleaver showed. He began by looking at the genes that are responsible for coding these regulatory receptors. There's a whole family of nuclear receptors, some of which are highly specific and classical, which mediate the activity of steroid hormones of various types, thyroid hormone, and the like. But the human genome contains dozens of other such receptor encoding genes whose functions had not been known and were called orphan receptors. Marching through this, he and many other laboratories have begun to identify the function of these orphan receptors, and he focused on one that's a receptor for pregnane. He showed that this particular receptor protein is expressed in the liver, small intestine, and colon by doing a northern blot hybridization for the RNA encoding this protein, and not in other tissues, so the locus of expression was in the right place. The next thing he had to do is show that this receptor can bind to cognate response elements in the promoter for CYP3A, and he did that by doing a gel study, an electrophoretic mobility shift assay, where you ask whether a particular protein can stick to a small piece of DNA. The small piece of DNA migrates very quickly in an electrophoretic field, an electrical field through a gel, but if a protein sticks to it, that larger complex migrates more slowly and is retarded. He was able to show that the PXR receptor can stick to this sequence in the promoter for CYP3A, but it does so and retards it only in concert with another member of the family. So the regulation of this is through a heterodimeric nuclear receptor process. The question then was whether drugs, including extracts of St. John's wort, working through by activating PXR, is capable of increasing the expression of CYP3A. Cleaver did this by using a simple reporter-based assay and showing in vitro that drugs known to upregulate CYP3A expression, like this tuberculosis drug, rifampicin, upregulated. So that's a good positive control. He then sent his technician to the local health food store and bought 3 different brands of St. John's wort, extracted those, added the extract into the same assay, and asked whether those also could drive the expression of this gene, and they did. They were then able to chemically separate dozens of components of St. John's wort and ask which of the chemical constituents of St. John's wort is capable of upregulating PXR activity and CYP3A expression. And of all of those, the drug that I mentioned before, hyperforin, which is a key component, we believe, of antidepressant action of St. John's wort, was exactly the moiety that activated this cascade. He went on to show, using primary liver cell cultures, that not only did hyperforin upregulate CYP3A, but it turned on a whole cascade of things dependent upon PXR. Other oxidating enzymes, conjugating enzyme, and transporting enzyme. The end result is this now explains the molecular and biochemical basis by which a potential herbal antidepressant would interfere with drugs. Now, drug companies are doing this today, and studying mainstream medications. We're understanding these interactions. If we're going to use herbal products for health purposes, we have to study them the same way. Let me move briefly to a second herbal product to talk about a different research strategy. And this is an herbal mixture known as PC-Spes, which stands for prostate cancer, and spes, I'm told, in Latin means hope. This is a cocktail of 8 herbs, chrysanthemum, licorice, ginseng, saw palmetto, and others. And it's being used today by over 10,000 American men with advanced prostate cancer, many of whom are hormone refractory. There have been 4 or 5 small studies, all of which have not been blinded, suggesting that this herbal remedy reduces pain, improves quality of life, and statistically significantly reduces prostate-specific antigen levels in the blood. If it's true, this would be remarkable. De Paola and his colleagues at Rutgers a couple of years ago showed that at least part of the activity of PC-Spes is because in the herbs are botanical equivalents of animal estrogens. And at least some of this is due to estrogenic activity, as evidenced by the fact that most of the men on this have a decrease in libido, have breast tenderness enlargement, and other estrogenic type responses. Adrienne Dobbs and her colleagues at Johns Hopkins University, through peer review, was funded to do the first placebo-controlled trial of this herbal remedy. This is in 100 men with hormone refractory disease, comparing it to a standard estrogenic treatment, estradiol. She's doing this in Baltimore and in Singapore, and measuring the kinds of things one would need to show that a prostate cancer drug is effective. But at the same time, we're learning how this product may work. Peter Nelson, who's a medical oncologist at the Fred Hutchinson Cancer Center in Seattle, is looking at the very genes that are regulated by the constituents of PC-Spes. Here he's doing an in vitro assay where he's growing prostate cancer cells, the LINCAP cell line, in the absence or presence of an extract of PC-Spes, extracting RNA from that, and binding that RNA to a gene microarray and asking what genes are upregulated and what genes are downregulated. He's already identified a couple of dozen genes, many of which are outside the androgen and estrogenic-dependent pathway, that shows that there's a broader range of activity than mere phytoestrogens. And he hopes to be publishing this in the coming months. So our work on dietary supplements covers a whole range from preclinical to phase 3 trials. At the same time, we were asking whether products are effective, we were asking how they work, and we're asking what kind of interactions they may have with other products. I'm going to turn now to talk about a second tier of study, which again are at the higher quality range within this field. And this deals with the area of mind-body medicine. We've known from physiologic studies for decades that various forms of meditation can slow the pulse and respiration, and as Herb Benson in Harvard called, induce the relaxation response. The question exists is whether these physiologic changes are clinically useful and how they are mediated. And I'm going to illustrate the beginning answers to those questions with a couple of sets of experiments. The first has to do with hypnosis. This is not new. Hypnotism was a parlor trick invented by Anton Mesmer in France in the 18th century. And Benjamin Franklin was a part of an international group of individuals who was brought in to challenge this and figure out what the trick is. And it led to a real questioning of the inherent basis of hypnosis. Nonetheless, over the past century, hypnosis has evolved as a practice for certain conditions. And contrary to what this cartoon suggests, that hypnosis involves putting people to sleep, it actually involves inducing a state of intense focus on a suggested response. And it's believed that that focus can induce involuntary changes in perception, mood, memory, or physiology. Many people thought that the hypnotized subject merely wants to satisfy the hypnotist and will go along with this little this little parlor trick. But there's some recent evidence suggesting that there's more to it. To tell you this story, I'm going to go through a bit of a complex experiment conducted by David Spiegel, who's a psychiatrist at Harvard, and his collaborator Steve Kostelin and colleagues in Boston. And they studied a model system and showed that hypnosis can alter how the brain processes color vision signals. Let me walk you through this. About 10% of people are highly hypnotizable. They took 8 such individuals and showed them identical patterns in color or in grayscale while they were undergoing positron emission tomography of the brain, PET scanning, which looks at functional changes in the brain, in this case, local changes in blood flow. The patients under hypnosis or without hypnosis were randomly told to see color or grayscale, regardless of what they were seeing. And the blood flow was shown to the various parts of the brain that relate to color processing, was shown to increase when the subjects were asked under hypnosis to perceive color, and decreased when they were told to see grayscale, regardless of what they were actually seeing. As David Spiegel likes to say, believing is seeing. This is a PET scan illuminating blood flow in the region of the occipital cortex involved with color vision, and other neighboring regions are more primarily associated with black and white vision. So you can ask, what is the brain working on, not just what is the brain being asked to see. And this is the pattern with identical gray tones, color or black and white. This is the data that I'm going to walk you through. We're graphing here the regional cortical blood flow in this part of the color association region, in the left cortex. And this is the amount of blood flow in an individuals not being hypnotized or hypnotized. Without hypnosis, people who are seeing gray and told to see gray had this basal level of blood flow in that color region. If they were seeing color, if they were seeing gray, if they were seeing gray and told to see color, it didn't change. They knew they were not seeing color. If they were seeing color, whether or not they were told it was color or gray, they had a higher level of blood flow appropriate for color vision. What happens now with hypnosis? If you're seeing gray and you're told you're seeing color, the blood flow went up to that of the level of color perception. And if you're seeing color and you're told under hypnosis you are to see color, the level went up even further. This suggests that the brain is acting as if what you're told to see is what you're seeing. There's a whole series of such techniques involving detailed brain imaging. Another set of such studies I'm going to come back to in a few minutes, but I'm going to detour briefly to talk about another area where the mind and body interrogate each other for surprising outcomes. And this has to do with the placebo effect. Now, historically, the placebo was an inactive or innocent contrivance that people used to encourage healing in the absence of something better to do. In research, we began to use a placebo to control for the nonspecific effects that might confound the calculation of the true benefits of a novel intervention. If you believe it's going to work, that may contribute somewhat to its effectiveness, just like if you believe you're seeing color, that might contribute somewhat to what you're seeing. Now, this is not new, and Thomas Jefferson talked about practices of his day in this fashion. He said, "One of the most successful physicians I've ever known has assured me that he used more bread pills, drops of colored water, and powders of hickory ash than all other medicines put together," which at that time was a good thing. But the modern age of the placebo was really marked by its use in research in the past 70 or 80 years, and by the seminal study by Henry Beecher of Harvard in 1955, who summarized 15 placebo-controlled trials, including an aggregate of over 1,000 individuals, and calculated the aggregate benefit from placebo alone was about 35%. And that figure of about a one-third improvement due to placebo became gospel in placebo-controlled trials in the past half century. And many of us who design studies assume that the placebo recipients can improve by a third, and if our drug is going to do better, it has to be statistically significantly better than 1/3. Well, this summary held sway until this past year, when Robertson and Gutcha of Denmark published in the New England Journal their version of the same analysis involving 116 trials with 8,500 subjects. Their conclusion, unlike Beecher's, is the placebo is not powerful, it's powerless. In the aggregate, they concluded that placebos have no beneficial effect, but were willing to admit that it was somewhat beneficial for subjective things, and particularly pain. Well, those of us who actually do these kinds of studies were absolutely flummoxed by this observation, and it was well summarized by the physician Sherwin Newland, who said that such a report could hardly negate an experienced physician's awe at a phenomenon that might impress even a dispassionate biometrician should he ever venture within the range of a real patient. Well, there are ways to study the placebo, and within the past couple of decades, there have been remarkable results of these. It turns out that about 2 decades ago, Howard Fields and his colleagues reasoned that if a placebo could relieve pain, it might be doing so by acting through the very pathways in our brains that control pain, involving endogenous opioids. Morphine, a plant narcotic, a plant opioid, merely provides an outside source of things that are already circulating within our brain. They reasoned that if placebos relieve pain through opioids, if you block opioids, you might block placebo activities. And they showed that a drug that blocks morphine action, a drug known as naloxone, completely eliminated placebo effects to pain. Fabrizio Benedetti in the University of Turin in Italy took that experiment one further, and I'm going to have to walk you through a very complex experiment here, that showed that not only does placebo analgesia work through this endogenous opioid system, but that it's specific for the part of the body. You don't just release these opioids through the brain, washing the brain like you would get a shot of morphine. It affects pain locally. And here's what he did. He took volunteers, Italian volunteers, and injected under their skin of all 4 limbs an extract of the chili pepper, an extract known as capsaicin. Now, for the same reason your mouth burns for several minutes after eating some hot salsa, their skin will burn. It'll take several minutes for that pain to diminish. They were told that randomly they'd be assigned to having a very powerful anesthetic cream applied to that site before the injection on one limb, not the others. And it turns out that the analgesia was achieved only in that limb, and the analgesic effect could be blocked by this narcotic blocking drug, naloxone. Now let me walk you through this. This is a simplification of a lot of tabulated data of the pain score and the disappearance of pain for all 4 limbs over time. Now, if you're going to inject this capsaicin, you're going to have pain, but if you preceded it by a placebo cream instead of an anesthetic cream, what will happen? The person will expect that there'll be perhaps an attenuation of the pain in that limb, or if it's in the leg, in the other limb. What really happens is that the pain diminished and went away more quickly in the very limbs that had the placebo cream applied, but not the other limbs. And if before the cream and before the skin injection you got a shot of placebo, it didn't affect it. But if you got a shot of naloxone, what happened is that relief of pain completely disappeared. It works through opioids, and it's spatially specific. Now, the latest saga, part of this saga, relates to this picture in Science Online 4 weeks ago, which the editors captioned, "This is your brain on placebo." This, again, is a PET scan, and what was done here is to take volunteers and to put a hot metal plate on the back of their hands to induce tolerable pain. They were given a shot of a narcotic analgesic or a shot of a placebo. And the parts of the brain that were activated in terms of regional blood flow by the narcotic or the placebo were mapped by PET scanning. It turns out that the region in the pons and the regions in the rostral anterior cingulate cortex were activated by both, that these regions completely overlapped. And placebo and opioid analgesia not only use the same biochemical pathways, but use the same neuronal pathways. So what have we concluded about the placebo? It can relieve pain. It works through the opioid system. It's anatomically specific and uses the same networks. We have to use these techniques for things other than pain, and they're being done now with treatment of depression. There was an article in Science 2 months ago showing similar results in treatment of depression with quantitative EEGs of the brain, and it's being done with Parkinsonism using deep brain stimulation, which relieves the rigidity and the tremor of Parkinson patients, and remarkably, so does a placebo. Now, it's the opportunity to use these powerful technologies in this important area of the placebo as it relates to mind-brain communications that led 17 institutes and centers and health agencies of the federal government in November 2000 to host a 2.5-day workshop on the placebo. It led to a book published this past week by British Medical Journal Press, edited by Harry Guess, Arthur Kleinman, our colleague John Cusick from NIDDK, and my colleague Linda Engel from NCCAM. But more importantly, it led to the issuance of RFAs that is on the street, and we are currently scheduled to receive applications that will be funded by 10 institutes to study the basic mechanisms underlying placebo effect, as well as how the placebo effect arises in the context of clinical practice. The answer to the research is a very practical one. It provides evidence, and the evidence leads to understanding. I've illustrated briefly that our work on dietary supplements have shown us that the potential benefits of dietary supplements may be offset by untoward drug interactions. We can study benefits, we can study interactions. And the mind-body dialogue that I've talked about in the context of meditation, hypnosis, and the placebo can yield powerful, even surprising physiologic responses. We shouldn't discount the potential of that to be harnessed for other clinical benefits. Ultimately, for the American public, this understanding leads to acceptance. We believe that this field of complementary and alternative medicine and its practices will be integrated with conventional medicine only when science affords a fuller understanding of its benefits of risks. And I have the placebo, there it is, I have the privilege of participating in that enterprise. Now, this issue of integration is here today, as well illustrated by this recent cartoon from the Washington Post, Baldo. Here's a dutiful mother caring for the sick child, and the father, assuming his role, is calling the doctor, saying, "Yes, doctor, Baldo is really not feeling well. His stomach hurts and he's coughing," and the thoughtful, truly wise mother says, "Sounds to me like he needs a little chamomile tea." The irritated conventional practitioner, the father says, "Tia Carmen, please, I can't hear the doctor." And the doctor says, "Have you heard of chamomile?" This is integrated, and our process of bringing to the NIH community some of the research finding that I've been able to summarize today will be continued in a distinguished lecture series that I invite you in the NIH community to nominate true distinguished people for. Charles Rosenberg, a professor of history of science, will be speaking in July, and Arthur Kleinman will be back here this coming November. This is what NCCAM is supposed to be doing. Thank you very much. Pleasure. Thank you.
So we have time here for questions. Please go to the microphone so that everyone can hear them, including the audience which is beyond this room. Let me start with a question about the study on the effects of St. John's wort on induction of transporters and detoxifying
I knew I'd
enzymes.
be in trouble. Michael's an expert in that field.
Assuming that St. John's wort or some of its components are shown to have some moderate antidepressant effect, the studies you describe suggest a hypothesis that some of those effects may be, in fact, because of the detoxifying effect that it induces. A prediction then would be that other agents, like rifampicin, that induce the same systems, might be antidepressants. Is there any data about that?
There is no data about that. It's a good question. Actually, there are several things. First of all, we're not sure that hyperforin is the active ingredient. And if it were not a really important constituent to the antidepressant activity, you could perhaps make an iteration of St. John's wort in which you've removed the hyperforin and got rid of that drug interaction. I am not aware that rifampicin has any beneficial effects for depression. My depressed patients on tuberculosis haven't woken up from it.
They do take it for a long time.
They do take it for a long time, yes.
Just a question about meditation. I wanted to ask you if there's any studies ongoing now for studying the good influence of the transcendental meditation, which are going now at NCCAM or NIH.
Yes, so transcendental meditation is one of many forms of meditation. There's actually an institution in Iowa that studies transcendental meditation. Interestingly, it's known as the Maharishi University. And that university is funded by NCCAM to do studies of transcendental meditation for hypertension and other cardiovascular conditions.
Could you comment on do you have any other initiatives together with the FDA or other regulatory agencies on these studies, and how can this new information be disseminated to the general public, and how that relates to the regulation of this industry?
So I'll try to answer that in two parts. The first thing is, if you look at our website, nccam.nih.gov, you'll see everything we fund, as well as a list of all of our initiatives. We've mounted about 27 initiatives since I became director in 1999. And I've just highlighted one of many. We at the NIH have no role in regulation other than to help provide the basic information on which regulatory agencies like the FDA could make more informed decisions. Dietary supplements are not regulated like drugs. They are available to the public, and the regulatory setting does not require proof of effectiveness or proof in a formal way of safety. We have worked closely with colleagues in various divisions of the FDA to talk about this process and how we could use more efficiently our investigative initiatives to develop the kinds of data that the public needs.
Could
I ask one question?
There's a question.
Does NCCAM have any information on people getting insurance coverage for alternative therapies?
Do we have any information for insurance coverage? You could call our toll-free clearinghouse, and there'll be information, I think, in the brochures outside that could help you get that kind of information, and that's the best way to answer it.
I was thanking Steve for ushering in a new era of study of complementary alternative medicine.
Think Forward. Think Research Channel.